Hi, everyone. Today, we want to discuss the role of myelin in neurological systems. So, what exactly is myelin? At its core, it's a highly modified cell membrane that forms a multi-layered sheath wrapping itself around nerve axons. This intricate structure is absolutely fundamental to the function of the vertebrate nervous system, where it acts as a biological insulator. In this explainer, we'll walk through a few key areas. We'll start with myelin's function as an insulator, then compare its formation in the central and peripheral nervous systems. After that, we'll dive into its unique molecular composition, focusing on the key lipids and proteins. And we'll finish up with a look at where myelin research has been and where it's headed. Okay, so first, let's examine myelin's primary role as an electrical insulator for the axon and discuss how its very structure facilitates this critical function. The impact of this insulation is a dramatic increase in the speed of nerve impulse propagation. As you can see, a myelinated axon can conduct signals up to 100 times faster than an unmyelinated one. This rapid transmission is achieved through a process known as saltatory conduction, where the nerve impulse essentially leaps between the unmyelinated gaps, which are called the nodes of Ranvier. Now, to quantify the degree of myelination, researchers use a metric called the G ratio. It's a fairly straightforward geometric relationship. You take the diameter of the axon itself and divide it by the total outer diameter of the axon plus its myelin sheath. It turns out there's an optimal value for this ratio, which differs slightly between the central and peripheral nervous systems. This highlights a very delicate balance. The sheath needs to be thick enough for effective insulation, but not so thick that it wastes space and metabolic resources. Maintaining this optimal thickness is crucial for efficient nerve conduction, and deviations are often associated with abnormal neural development and various neurologic diseases. So we've discussed what myelin does. Now let's look at the cells that actually build it. Myelin is produced by two very distinct types of glial cells, and which one is used depends on its location in the nervous system. In the central nervous system, or CNS, oligodendrocytes are the architects. A single oligodendrocyte has the capacity to myelinate segments of 40 to 60 different axons. In contrast, in the peripheral nervous system, or PNS, Schwann cells do this job, and each cell dedicates itself to just one segment of a single axon. This table also points out other differences, like their key structural proteins and unique features, such as the Schmidt-Lantermann incisors you find in the PNS. These architectural distinctions help explain why certain diseases selectively target either CNS or PNS myelin. We'll now shift our focus from the cellular to the molecular level. Myelin has a highly specialized composition that really sets it apart from other biological membranes. For context, a typical cell membrane maintains a relatively balanced ratio of proteins to lipids. It's about a 50-50 split by dry weight. Myelin, on the other hand, is dramatically different. It's characterized by an exceptionally high proportion of lipids. We're talking 70 to 85% of its dry weight. This lipid-rich nature is fundamental to its function, making it less permeable to ions and, therefore, a superior electrical insulator. This molecular makeup gives rise to a very distinct ultrastructure. When you view it under an electron microscope, you can see two repeating features. The major dense line is formed by the tight fusion of the inner or cytoplasmic surfaces of the membrane. And then you have the intraperiod line, which is slightly wider, formed by the apposition of the outer or extracellular surfaces. Given their sheer abundance, it's clear that lipids are the primary building blocks of the myelin sheath. So let's take a closer look at the specific classes of lipids involved. The lipid composition of myelin is quite distinct from a typical membrane. You can see it contains a significantly higher proportion of cholesterol and about a twofold increase in glycolipids. The overall ratio of the major components is approximately 40% cholesterol, 40% phospholipids, and 20% glycolipids. Each of these lipid classes plays a specific important role. The rigid structure of cholesterol, for instance, increases the packing density of the membrane. The phospholipids in myelin have long hydrophobic tails that promote tight association and improve insulation. And then the glycolipids, through extensive hydrogen bonding, help create dense, rigid domains that make the entire membrane more stable. Although they are less abundant, proteins are the molecular engineers that organize and compact these lipid bilayers into a functional sheath. We're going to focus on three of the most important ones. So first up is proteolipid protein, or PLP. This is the most abundant protein in CNS myelin. It's a highly hydrophobic protein that spans the membrane 4 times. Its primary job is to bring the extracellular surfaces of the myelin membranes together, which forms the intraperiod line. Mutations in the gene for PLP are known to cause Pelizaeus-Merzbacher disease, which is a severe hypomyelinating disorder. The second most abundant protein in CNS myelin is myelin basic protein, or MBP. Now, unlike PLP, MBP is what's known as an intrinsically disordered protein, which means it lacks a fixed 3-dimensional structure. This gives it a lot of flexibility. It's found on the cytoplasmic side of the membrane and carries a high net positive charge. This allows it to act almost like an electrostatic glue, binding the negatively charged cytoplasmic leaflets of the membrane together to form the major dense line. So in the CNS, you have PLP and MBP working in concert to compact the myelin sheath. PLP handles the extracellular surfaces, while MBP takes care of the cytoplasmic surfaces. In the peripheral nervous system, however, this dual role is handled by a single dominant protein, myelin protein 0, or P0. P0 is the major protein of PNS myelin, making up over half of its total protein content. It's really a multifunctional molecule. Its extracellular domain acts as a homophilic adhesion molecule, which just means it binds specifically to other P0 molecules to hold the extracellular surfaces together. At the same time, its positively charged cytoplasmic tail performs the same function as MBP in the CNS, compacting the cytoplasmic surfaces to form the major dense line. To conclude, let's briefly review the history of myelin research and highlight some of the key questions that are driving the field forward today. The study of myelin has a long history, starting with its first observation all the way back in 1717. Key milestones include it being named by Virchow in 1854, Ranvier's identification of the nodes in 1878, and the confirmation in 1962 that oligodendrocytes are the source of CNS myelin. Today, the field is shifting from viewing myelin as just a static insulator to seeing it as a dynamic system. A key area of research is adaptive myelination, which explores how neural activity and experiences can actively shape the structure of myelin, suggesting a role in learning and memory. And despite all this extensive research, many fundamental questions remain. For example, while we understand the components of the nodes of Ranvier quite well, the mechanism that regulates the distance between these nodes is still unknown. As an axon grows, this internodal distance has to change accordingly. But how that information gets conveyed to the myelinating glial cells is still an open question, and it really highlights the complexity that continues to drive discovery in this field.